Welcome back. This is the 7th lecture in our Chapter 2 series. We've gone all the way through subatomic particles and bonding to organic molecules like carbohydrates, lipids, proteins, nucleic acids. Make sure you know the monomers and polymers of each. Okay, just as a quick review, we've talked about carbohydrates and the monomer being monosaccharide. We've talked about carbohydrates and the polymers, both in plants and animals, as being starch, cellulose, and glycogen. Remember the disaccharides and the examples of the monosaccharides and disaccharides discussed. Make sure you know lipids and the different types, triglycerides being the main form of long-term energy storage, and phospholipids being the main source of membrane construction. Make sure you know proteins and the amino acid monomer or peptide monomer and how those monomers are linked together with a synthesis reaction like dehydration synthesis to produce polypeptides and proteins, and how nucleic acids are composed of the monomer nucleotides. Okay, but now we're into enzymes, and if you've heard me say in class, enzymes are part of that protein, part of the protein group of organic molecules, and so we're kind of going back to the proteins in terms of like the monomer and polymer, but enzymes are specific proteins or specific tertiary and quaternary structures that allow you to do things. And enzymes, like this depiction say, are everywhere. Okay, all life requires the use of an enzyme, and I know I say this all the time in class that every single biological reaction requires the use of an enzyme. There isn't a biological reaction, and what I mean is a reaction in life or a reaction that a life form depends on. All of those require the use of an enzyme. Okay? There isn't a plant, animal, protist, fungus, bacteria that doesn't use an enzyme. And we're going to see later how enzymes help our cells. Enzymes are biological catalysts, okay, which means they're going to do a couple things for our cells, and we'll get to it in a minute. Okay, but enzymes are everywhere. We're going to get into why enzymes are the catalyst, or how enzymes act as a catalyst. And the two main things that allow enzymes to be considered biological catalysts would be in the area of reaction rate. Enzymes are going to allow us to speed up our chemical reaction. Enzymes speed up chemical reactions and they lower activation energy. Okay, lower activation energy, and as we get to in a minute, is the amount of energy it takes to start or initiate a reaction. And so enzymes make it easier for reactions to take place, and because they make it easier, they speed it up. So these two things go hand in hand. Making an energy reaction or making the activation energy lower is going to speed up the reaction because you're decreasing the amount of energy it takes to get it started. So naturally you should speed it up. But let's get into a little bit about enzyme structure. Enzymes are biological catalysts, but they're quaternary proteins, and we've talked about it in class that quaternary proteins are made up of multiple tertiary proteins, so it's a globular shape, which are made up of those alpha helices and pleated sheets. And when you put a bunch of tertiary proteins together and produce a globular protein that produces an enzyme, there is a spot or a location that is kind of like a piece of a puzzle that has kind of a cutout, if you will, and this substrate is going to bind to that spot perfectly. Okay, the substrate has to fit in the active site perfectly, and that brings up a term that we need to talk about called substrate specificity, or enzyme substrate specificity. And that really just means that there is a specific substrate for every enzyme, meaning every enzyme acts on a different substrate. Okay, in this case, in this example, our substrate is sucrose, and sucrose has been talked about a lot in class. When we talked about carbohydrates, remember sucrose is a disaccharide, okay, and you can see that. Here's one monosaccharide, and here's two monosaccharide, and they're linked via dehydration synthesis, okay, to produce a disaccharide, in this case sucrose. That substrate is going to fit into this active site perfectly in order for this enzyme substrate complex, which can be seen here, in order for this thing to form. Once this enzyme and substrate bind, they're going to produce this enzyme substrate complex. And if the enzyme, in this case, is in charge of breaking the bond between the first monosaccharide and second monosaccharide, it requires the use of water. Okay, this is a hydrolysis reaction. We're going to split water into an H and an OH, and when we split water into these two things, we're going to give one to one of them, and we're going to give the other one to the other one. And because we're giving these two substrate parts or components something else to hold on to, they break the bond between them and become individual. Once they are individual, they will be released as two individual monosaccharides that the cell can now use in terms of energy. Okay, so enzymes allow reactions to happen because they lower the amount of energy it takes for that reaction to take place, and it speeds it up by reducing the amount of energy it takes to take place. Okay, a couple things. Substrate complex or enzyme substrate complex means that we have bound our substrate and enzyme together, and enzyme substrate specificity refers to the fact that there is a specific substrate to a specific enzyme, and the active site has to fit the substrate perfectly. One thing we talk about is a lock and key analogy or a lock and key hypothesis. This will be one of the essay questions you'll see on your test, and it refers to just like a lock and key. Okay, you have a lock, which is the substrate, you have the keyhole, which is the active site, and you have the key, which is the substrate. And I don't know how much experience you have with keys and locks, but you know at this point in your life that a specific key has to fit a specific lock. I can't stick any key into any lock and expect it to work. Enzymes are the same way. We can't stick any substrate into any enzyme and expect that enzyme to work or act upon that substrate, because that's not the way it works. We are going to put a specific substrate into a specifically shaped active site in order for that enzyme and substrate to bond, in order for that enzyme to produce the products that that enzyme is supposed to produce. Okay, that's a lock and key analogy. One thing also we're going to talk about or kind of rehash is this enzyme substrate complex and enzyme substrate specificity. Again, it's an enzyme is specific to the substrate it binds to, and the active site has to fit the substrate perfectly. Okay, I know I've said this like 3 times, and you're probably getting tired of hearing it, but it's one of those really, really important concepts of this entire lecture. Enzyme substrate specificity. When an enzyme binds to a specific substrate, it's going to cause the enzyme to change shape, and that's really important because it allows an energy transfer to take place. Because every time we break a bond, we release energy. Bonds in science, or bonds in chemistry, bonds in molecules, always store energy. And so if there's a certain amount of energy stored in this bond, when we break the bond, we're going to release energy, and it's going to transfer either into our enzyme into something else, or it's going to be released into the cell, most of the time in the form of ATP. Okay, again, this is a hydrolysis reaction, or a synthesis reaction rather. We are taking a larger polysaccharide or polymer, and we're breaking it down into individual monomers. So that requires water. That is a hydrolysis type reaction. A little humor for you. They call me DJ Enzyme because I'm always breaking it down. Get it? Enzymes are always breaking larger molecules down. Okay. Another one for you. I can't remember the name of the enzyme that breaks down carbohydrates. It's right on the tip of my tongue. Because your saliva has enzymes in it. Okay. Let's talk a little bit about the type of enzymes, and specifically competitive and non-competitive in inhibition. If we talk about enzymes, remember I've already talked about there is a substrate that is specific to the enzyme in which it acts on. The enzyme has a specific shape cutout that the active or the substrate fits into, and that active site has to fit the substrate perfectly, just like the lock and key analogy. The key has to fit into the keyhole, and fit that lock perfectly in order for that lock to open or unlock. And so enzymes are the same way. Enzymes have a specific substrate that binds to a specific shaped active site perfectly in order for that enzyme to function. And that enzyme could be in charge of taking oxygen from the lungs to every single cell in your body. It could be an enzyme that's in charge of the breakdown of glucose into ATP. It could be an enzyme in charge of breaking down complex carbohydrates like glycogen into individual monosaccharides like glucose. All enzymes have a specific role, and these are things that affect the ability of an enzyme to act like it should. Okay, temperature is really, really important. Why is it important that you maintain a constant internal body temperature? Many of you would say that it's part of the homeostasis that makes us us, right? We have to contain or continue to be at a constant 98 degrees. If our temperature fluctuates above or below even a couple degrees, you can start to pull these quaternary proteins that make up these enzymes apart. And on the next slide, I'll show you kind of an image that shows this denaturation process. But temperature is really important. Why is a really high fever or heat stroke life-threatening? And the answer is high temperatures, 105, 106, 107 degrees, core internal body temperature can cause all of your enzymes that are made up of proteins to denature or pull apart. And if you remember anything about structure dictates function, those enzymes have to be structured that way to function correctly. And if you start changing the way they're structured, meaning you're changing the shape, they're not going to function correctly. And if they don't function correctly, you're in trubs, right? You can't survive. Okay? As an organism and as a cell, you're in trubs. Enzyme concentration. If we increase or decrease the concentration of those enzymes, it will impact how easily or how many substrates they have to act on. If you have a really low enzyme concentration, they have a lot of substrate to act on, but they can't keep up because there's not enough of them. If you have too high an enzyme concentration, then there's not enough substrate for them all to act on, and they kind of just get in each other's way. So you want to balance just like you have other homeostatic balances. Enzyme concentration is the same way. pH is really important also, as well as salinity. All of those things are able to affect enzyme concentration or enzyme activity. pH affects how easily the enzyme works. Okay, if an enzyme is in too high a pH or too low a pH, it can't function correctly. Competitive and non-competitive inhibition is like it shows. If this secondary substrate binds to the active site in place of the primary substrate, then it's going to block the primary substrate from binding, and it's not going to allow this enzyme to work, meaning it's going to inhibit the enzyme. Okay? It keeps these from bonding. That is a competitive inhibitor. Non-competitive inhibitors are going to bind to the secondary active site, and because it wedges its way in here, notice that the opening of this active site is constricted because we kind of rotated all of this mass this way. And if we rotate it all this way and we keep this active site from being the shape it should, or we change the shape of this, then all of a sudden the active site that is supposed to fit the substrate perfectly no longer fits. And again, you're inhibited. But this is non-competitive inhibition because we're not competing for the same spot. Here's a picture of the denaturation process. Notice that a normal protein, like discussed in class, is composed of sections that are alpha helix and pleated sheet, which can kind of be seen. It's not really good, but these kind of in here would be the pleated sheet. It's more wavy, but it should be more folded accordion shape. But we're made up of multiple secondaries. It is overlapping, so this would be a normal quaternary or tertiary protein, depending on if it's more than one. When you increase the temperature or decrease the temperature, the protein and the hydrogen bonds holding these folds and coils together no longer hold on to each other. And so if all the hydrogen bonds release their grip, or you break those hydrogen bonds, you pull this protein apart. And a protein that is supposed to be structured this way no longer has the same function because it's now structured this way. Again, like I said before, you'd be in TRUPS. So here is a space for me to put in the enzyme function. Make sure you can graph this. We're going to start with normal x and y axes. If I can change the ink color and look at a graph curve that, come in here, a graph is going to appear to be like this. The starting point is going to be A, the ending point is going to be B, and the x-axis would be time, and the y-axis is going to be energy level. As energy increases, time increases. Okay, so that's how we're setting it up. This is a normal reaction rate. You always start with the reactants at A. These are our starting products, okay, or our starting reactants. These are the things that are going to react. They require energy or an input of energy to this point right here. And at this point, no more energy is needed because at this point, if you think about gravity, if this was a hill that you climbed and or a hill you're going to sled down or ski down, at this point gravity would take over and the rate of reaction would take place without the input of any additional energy. So if we were to look at the distance from starting to ending, this is activation energy. Okay, activation energy. This is the energy it takes to activate the reaction. Well, what happens when you add an enzyme? Enzymes are going to start at the same reactants because we're using the same reactants. It's the same reaction, but this time we're going to add an enzyme, and so what did it do? It lowered the activation energy. Okay, this is the activation energy of the reaction with enzyme. So red is with enzyme, and the blue is the reaction without enzyme. So what's the difference? No enzyme. The difference is the amount of activation energy. The reaction without an enzyme uses almost twice as much energy to get this reaction started. If we were going to ski these mountains, would you like to hike this mountain or would you like to hike this hill if we could get to the same point in B? Now you might say this first hill would be more fun, it'd be steeper, like it's longer, the skiing would be longer, but think about the amount of work it takes plus where we want to go. I think it would be better to just hike this one and then still get to the same place, right? And so why do enzymes matter? They are the things that lower the activation energy and speed up the reaction. We would get to the B point here by traveling the enzyme reaction faster than if we were traveling the without enzyme reaction. Let's talk a little bit about synthesis and decomposition. We've already started talking about in class hydrolysis and dehydration synthesis. Notice that synthesis is a reaction that requires energy and you are always building up larger. So you're going from small to larger. Okay, we are putting one monosaccharide together with another monosaccharide by pulling out water to build a disaccharide. If you were to contrast that, this one, decomposition, is going to release energy sometimes in the form of ATP, or you're going to release it as heat, and these are your hydrolysis reactions. So this would be taking a larger molecule, like a disaccharide, and splitting them up into smaller monosaccharides. Okay, this one means we have to put in water, because we're going to split water to give one of these an H and one of these an OH, so we're going to split them back up into individual monosaccharides. This one requires the removal of water. So, synthesis reactions, dehydration synthesis is a synthesis reaction. Its job is to remove water in order to build up larger molecules. Hydrolysis reaction is a decomposition reaction. Its job is to put in water to break larger molecules into smaller molecules. What do you call the enzyme that breaks down ice cream? Haagen-Dazs. Why is that funny? because enzymes end in ace and it sounds like Haagen-Dazs. There you go. From all of us here, I'm Mr. Pfeiffer. You stay classy, Washburn Rule. See ya!